So if you could somehow, I don't know, rewind the tape of life all the way back to the very beginning and just hit play again, would evolution repeat itself?
Right. Would we end up with the exact same biology or, a completely different story?
Exactly. Would we get the exact same creatures? It's a wild thought experiment.
It really is one of the most profound questions in biology. I mean, we tend to view evolution in hindsight as this strict march of progress, right? Like it has a predetermined outcome. But it raises this massive issue of determinism versus randomness.
Right.
Like, is evolutionary success just destiny driven entirely by the environment, or is it heavily influenced by just, a series of random, unpredictable accidents?
And usually that is strictly a philosophical question. We can't actually rewind a billion years of Earth's history to literally watch the dinosaurs roll the dice again.
Yeah.
But today we are looking at a stack of research where scientists figured out a way to perform this exact thought experiment.
Yeah, it's fascinating.
Not with entire ecosystems, obviously, but with your immune system.
Yeah.
So we are unpacking this incredible 2026 paper from the journal Cell, and it's titled Replaying Germinal Center Evolution on a Quantified Affinity Landscape.
Yeah, and the authors of this paper essentially built a microscopic evolutionary sandbox. They took the exact process that happens inside your body, every time you get sick or get a vaccine, and they just hit play on over 100 independent parallel evolutionary races.
Just to see how they would unfold.
Yeah.
So the mission for today's deep dive is to thoroughly unpack how our immune system actually learns to fight pathogens.
Right.
We're going to explore every mechanism from the cellular level way down to the genetic level without skipping a beat.
And the setup for this is it's highly specific.
Right. They didn't just use any cells.
Exactly. The researchers started with a single population of identical unmutated B cells. They called this starting point clone 2.1. And the specific B cell is naturally coded to target a model antigen called chicken IgY.
Chicken IgY, which is really just the bird version of an antibody, right?
Yeah, exactly. It just acts as a harmless foreign target here for the experiment. So to set up this controlled race, they took these identical clone 2.1 B cells and transplanted them into a very specific environment, genetically modified mice.
Okay, so the goal was to give these transplanted cells a completely empty arena. But how do you actually create an empty arena inside a living, breathing mouse?
Right. It's tricky. They engineered the mice using this classic genetic tool. It's called a CD23 Cre and BCL6 conditional knockout system.
Let's break that genetic modification down a bit for the listeners.
Sure. So BCL6 is a master transcription factor. It's basically a gene that acts like a master switch, and it's completely required for B cells to form a structure called a germinal center.
Okay.
Without BL6, a B cell simply cannot participate in the immune response. Period. So the researchers used a promoter called CD23, which is engineered to only turn on inside B cells.
Got it. So when it turns on, what happens?
When CD23 turns on, it activates the Cre enzyme. And you can think of Cre like molecular scissors. It's designed to specifically cut out the BCL6 gene, but only in the mouse's native cells.
Oh, wow. So the mouse's own immune cells are just genetically locked out of the whole process.
Exactly. Leaving the physical structure of the lymph nodes totally intact, but entirely empty of native competitors.
It really is an empty stadium. That is brilliant.
It is. And then they immunized these engineered mice with that chicken IgY antigen. This triggered a massive immune response, but the only players actually allowed on the field were those identical transplanted clone 2.1 B cells.
Right, because the native ones can't form the germinal centers.
You got it. And by doing this across multiple mice, they seeded 119 separate, completely isolated immune responses. So 119 parallel universes, all starting from the exact same baseline.
Just to see if evolution plays out the same way in each one. Okay, but before we look at the results of the race, I think we need to quickly map out the actual racetrack, the physical space where this is happening.
Yeah. Good idea. The action takes place in germinal centers, which form inside your lymph nodes.
Right, like if you've ever felt those swollen glands in your neck when you have a bad cold, you are actually feeling these germinal centers just swelling with activity. I like to think of them as ruthless, hyper-competitive boot camps for your immune system.
That's a really great analogy, because when a B cell enters a germinal center, it starts this high-stakes process called somatic hypermutation, or SHM.
And this sounds crazy to me. The cell actually mutates itself.
Deliberately. The B cell physically unwinds its DNA and intentionally introduces mutations into the genes that code for its antibody receptors, specifically the I and EEC genes.
See, the idea of a cell deliberately mutating its own DNA just seems incredibly dangerous. I mean, usually we associate DNA mutations with, radiation or cancer or disease,
Mhm.
how is the cell surviving that risk?
It's a calculated risk, and it's tightly restricted to just a tiny, tiny portion of the genome. The B cell uses an enzyme called AID.
AID.
Right, which stands for activation-induced cytidine deaminase. What this enzyme does is it chemically alters specific DNA letters. It basically converts cytosine into uracil.
Which shouldn't be there.
Exactly. The cell's natural repair machinery comes in, recognizes the error, but the repair process in this specific region is intentionally sloppy.
Oh, interesting.
Yeah, so that sloppy repair results in a permanent mutation. The B cell is essentially introducing these typos on purpose, hoping that one of them will change the physical shape of its antibody.
So it can grab onto the invading pathogen with a tighter grip. It's like taking a key, breaking it, and taping it back together over and over, just hoping it accidentally opens the lock better than the original one did.
That's exactly what it's doing.
But tracking millions of microscopic cells doing this inside a living mouse, that sounds completely impossible.
It would be, without some serious bioengineering. They used a photoactivatable GFP transgene to actually see it.
Okay, what does that mean? Photoactivatable.
Right, so the transplanted cells carried a gene for green fluorescent protein, or GFP, but it's photoactivatable, meaning it just sits there, completely dormant and invisible until it gets struck by a very specific wavelength of light.
Oh, so they can turn the glow on and off?
They turn it on using a multiphoton laser. This laser uses two low-energy photons that hit the exact same focal point at the exact same time. It lets the laser penetrate deep into the living tissue without, burning the mouse.
So they're literally playing laser tag with specific germinal centers inside a living animal. That is wild.
Yeah, it's incredible tech. So by pulsing this laser at day 15 and day 20 after the immunization, they permanently change the protein in those specific cells, making them glow green.
Which means they could extract the tissue and sort them.
Exactly, using flow cytometry, a machine that sorts cells one by one based on that fluorescence. They basically surgically plucked out the glowing cells. In the end, they sequenced 8,744 individual B cells from those 119 replicate germinal centers.
Okay. Let's pause for a quick recap for you guys listening. We've got 119 isolated boot camps. Every single one started with the exact same clone 2.1 genetic blueprint. Every single one is fighting the exact same chicken IgY target. So think about a time you tried to solve a complex problem with a large group of people. Did everyone independently take the exact same path to the solution? Because if evolution is just a predictable optimization machine, shouldn't every one of these boot camps arrive at the exact same, quote-unquote, best mutated antibody?
And the data gives us a resounding no. They absolutely do not.
Really? Completely different.
Completely. The researchers used all that genetic sequencing data to reconstruct phylogenetic trees for the B cells inside each germinal center. So think of these as massive family trees showing which mutations gave rise to which descendant cells.
Yeah.
The topologies, meaning the physical shapes of these family trees, were just wildly different from one germinal center to the next.
So how did they measure that? Like, how do you quantify the shape of a family tree?
The paper focuses on two specific metrics. The first is the normalized dominance score, or NDS.
Okay, NDS. And that's basically a measure of market share, right? Like if one specific mutated cell and its descendants end up making up 90% of the total population in a germinal center, that would be a very high NDS.
That is perfectly put, yes. NDS measures the sheer size of the largest lineage. Now, the second metric is the maximum recent expansion index, or REI.
REI.
While NDS measures overall size, REI measures the speed and the suddenness of the growth. It detects what we call a clonal burst.
Okay, so a clonal burst isn't just a cell steadily dividing over a few weeks. It's more like an explosion.
Exactly. A sudden, massive proliferation. It happens when a single cell hits the genetic jackpot. It finds a mutation that gives it such a severe advantage that it just begins dividing furiously.
Wow.
It completely overtakes the local environment and just starves out the competition within a matter of days.
So when we look at how these two metrics interact, NDS and REI, we get this fascinating picture of evolutionary diversity. Like some of these germinal centers look like highly branched bushes.
Right. Low dominance, low expansion.
Yeah, it looked like a diverse free market.
Yeah.
Dozens of different mutant lineages competing, but nobody is really able to gain a clear upper hand.
Exactly.
But then other germinal centers look like a single solid tree trunk. High dominance, high expansion. They operated as these ruthless monopolies where a single cell discovered a legal upgrade and violently wiped out the rest of the boot camp.
But here is what is truly remarkable about the overarching outcome. Despite these entirely disparate trajectories, some are messy and diverse, others are aggressive monopolies, every single one of the 119 germinal centers successfully increased its overall binding affinity to the target.
So they all arrived at a superior biological weapon, but they took completely different evolutionary roads to get there.
Yes, exactly.
Okay, so recapping this section for the listeners, we got NDS for market share, REI for clonal bursts, and we see both diverse bushes and monopolistic trunks. So I have to ask you, listening, which system do you think is actually better for long-term survival? A messy, diverse market of moderately good antibodies, or a highly optimized monopoly that risks putting all its eggs in one basket?
And that strategic tension really leads us to the next layer of the paper's analysis. Because if the immune system consistently finds a way to improve its grip, the question becomes, what specific mutations is it actually discovering?
Right, like out of all the possible choices.
To answer that, the researchers had to look beyond what the cells did choose and physically map out everything they could possibly choose.
Bees basically needed to build a complete map of the terrain.
Exactly.
And they did this using a technique called deep mutational scanning, or DMS. They took the original clone 2.1 antibody and created a yeast surface display library that systematically tested 4,158 single amino acid replacements.
Which is a huge undertaking. They essentially tested every conceivable single-step mutation that antibody could possibly make.
And for each of those 4,000 mutations, they measured two distinct physical properties, right?
Yes. The first is the change in affinity, meaning, does this specific mutation make the antibody stick to the target tighter, or does it weaken the grip? The second property is the change in expression, which is a way of evaluating folding stability.
Okay, wait, let's pause there. Why does folding stability matter just as much as the actual grip?
Well, proteins are incredibly complex, three-dimensional origami structures, and the sequence of amino acids dictates exactly how that origami folds.
Right.
So a mutation might create a shape that binds perfectly to the target, but if that same mutation disrupts the structural integrity of the protein itself, the whole antibody just unfolds into a useless string of amino acids, and the cell dies.
Oh, wow. So the cell constantly has to balance its function with its physical stability.
Yes, and when the researchers mapped out this delicate balance, the mathematics of survival proved to be brutal. They found that for every one single mutation that actually improved the antibody, there were 10 mutations that fundamentally damaged it.
10 to 1. So these B cells are just navigating an absolute minefield where the vast majority of random steps lead to total failure.
Precisely. And to comprehend why the failure rate is that high, the researchers used structural cryo-electron microscopy.
Cryo-EM.
Right. It involves flash freezing the protein complex in this ultra-thin layer of vitreous ice and then shooting electrons through it to build a 3D model at the atomic level. They wanted to see exactly how the clone 2.1 antibody was physically grabbing the CH2 domain of that chicken IgY antigen.
So if we visualize the paratope, which is the actual binding site on the antibody like a lock, and the antigen CH2 domain is the key, the cryo-EM imaging revealed something pretty surprising.
Yeah.
The central groove of this clone 2.1 lock is already a near-perfect fit for the key.
Exactly. And because that central interface is already physically optimal, any random mutation that happens in the middle of that groove just disrupts the established atomic bonds. It ruins the fit.
It makes it worse.
Right. So the only viable path for the antibody to improve its grip is by tweaking the peripheral edges of the paratope.
Right.
The beneficial upgrades literally only happen on the outer rim of the contact area.
And what's really cool is they found that these edge mutations are largely additive.
Yes, they stack.
Right, so if a mutation on the left rim provides a slight boost and a completely separate mutation on the right rim provides another slight boost, a cell that manages to acquire both will essentially add those benefits together.
Exactly. It's a landscape where one wrong step in the center drops you off a cliff, but tiny cumulative steps around the edges slowly lead you to the peak.
Okay, so recapping the deep mutational scanning. They mapped the terrain, found a brutal 10 to 1 bad to good mutation ratio, and discovered that because the center of the lock is perfect, you can only upgrade the edges. So visualize this treasure map in your mind, because that brings us to a critical junction. The map shows exactly which peripheral mutations yield the highest improvements. So the obvious question is, when we look at the actual cells pulled from the living mice, do they find these optimal mutations?
And the startling answer is no. They don't.
Wait.
Germinal centers completely miss the vast majority of the best possible upgrades. They leave massive amounts of binding power on the table.
But why? If the immune system is supposedly this great optimizer, why is it leaving money on the table?
It comes down to a concept called intrinsic mutability, and this really challenges a common assumption about evolution. We tend to just assume that if a trait is highly advantageous, natural selection will inevitably find it and preserve it.
Right, survival of the fittest.
But intrinsic mutability demonstrates that the likelihood of a mutation actually occurring isn't driven by how useful it is. It's driven by how easy it is for the DNA to physically mutate at that specific location.
Wait, hold on. You mentioned earlier that the AID enzyme is what causes these mutations. Are you saying the enzyme itself is biased? Like, how does a chemical enzyme prefer a specific part of the DNA? Isn't mutation supposed to be just a random scattergun approach?
It is entirely biased. AID does not bind to DNA randomly at all. Its chemical structure dictates that it binds much more efficiently to specific short sequences of DNA, which we call hotspots.
Okay, give me an example.
For instance, it strongly prefers a motif known as WRCY.
WRCY.
Right. If the DNA sequence matches that exact motif, AID will aggressively mutate it. But if a highly beneficial amino acid upgrade requires a mutation in a region of DNA that lacks these motifs, what we call a cold spot, the enzyme simply will not bind there.
Wow.
And the mutation almost never occurs.
It is literally like trying to win the lottery, but your biology fundamentally bans you from picking odd numbers. Like, even if the winning ticket is mostly 3s and 7s, you physically cannot play those numbers.
That's a perfect way to put it. And furthermore, the researchers observed a strict constraint involving the genetic code itself.
You mean the codons?
Yes. It takes a sequence of 3 DNA letters, a codon, to code for one single amino acid. The paper found that the germinal centers almost universally failed to find beneficial mutations if achieving that new amino acid required changing more than one nucleotide in a single codon simultaneously.
Oh, because the odds of hitting 2 right next to each other at the exact same time?
The statistical probability of AID hitting 2 specific adjacent letters at the same time is so infinitesimally small that the B cell is effectively locked out of that evolutionary pathway entirely.
That is wild. They tracked a B cell lineage that had been actively mutating in a germinal center for 10 weeks, which, by the way, is a massive portion of a mouse's entire lifespan. And even after all that time, it never found the highest value mutations because the spelling rules of its own DNA prohibited it.
Exactly.
So recapping this section, evolution isn't a limitless, perfect optimizer. It is heavily constrained by intrinsic mutability and spelling biases. So how does that change your view of evolution, knowing it's restricted by its own internal typos?
It's definitely a paradigm shift. So we've established how deeply individual cells are constrained by their DNA. But we have to zoom back out to understand how these constraints impact the entire population over time.
Right, the big picture.
The researchers consolidated all this data, the deep mutational scans, the tree topologies, the time points, and they constructed a mathematical model known as a fitness landscape.
A fitness landscape. Okay, so this maps out exactly how much a specific boost in binding affinity actually translates to a survival advantage for the cell, right? Like, does a tiny improvement give you a huge advantage, or does it barely move the needle?
Exactly. And the math revealed a remarkably clean biological relationship. They found that a 10-fold increase in binding affinity directly translates to a B-cell lineage doubling its population size relative to its competitors roughly once a day.
Wait, once a day? So if a cell discovers a mutation that makes it 10 times stickier to the target, it gets to divide twice as fast as the cell right next to it. That kind of exponential growth totally explains those massive clonal bursts we talked about earlier.
Absolutely.
But it can't just scale upwards infinitely, can it? Like, that has to be a limit.
There is. The mathematical model showed a very distinct plateau. The fitness advantage caps out when the antibody reaches an affinity of around 40 picomolar.
40 picomolar.
Right. Once a B cell hits that specific threshold of stickiness, getting even tighter doesn't confer any additional reproductive speed. The cell has just hit the absolute ceiling of the fitness landscape.
And understanding that ceiling leads us to what is, honestly, probably the most profound conceptual shift in the entire paper. It addresses this illusion known as survivorship bias.
Yes, this is crucial. If you read a standard biology textbook, it often describes germinal centers as somewhat permissive environments.
Permissive, meaning forgiving.
Yeah, the logic is that when scientists look inside a germinal center, they always find a mixture of elite, high-affinity cells living right alongside weak, low-affinity cells. So it makes the boot camp look like it has a soft spot for the underperformers.
But the paper completely dissects this illusion, right?
Yeah.
By identifying two distinct evolutionary forces that are just skewing our perspective. The first one is the push of the past.
Right. The push of the past is a classic phylogenetic trap. When we look at the evolutionary tree of a successful cell, its early ancestors appear incredibly fit, simply because they produced so many modern descendants.
But we are only looking at the lineages that survived to the end of the race. We are completely blind to the thousands of sibling lineages that were born at the exact same time but died out entirely. It's the ultimate example of history being written by the victors. The past looks perfectly engineered for success only because the failures were completely erased from the fossil record.
Precisely. And the second force, the pull of the present, explains why we see those weak cells currently residing in the germinal center in that snapshot.
Okay, so why are they there?
It relates to how the germinal center actually enforces its selective pressure. Inside the light zone of the germinal center, B cells must physically tear antigens off the surface of structural cells and then present those antigens to specialized T follicular helper cells.
So they have to literally show their work to a superior officer.
Yes. And if OB cell has a weak, low-affinity antibody, it fails to grab enough antigen. So when it meets the T cell empty-handed, the T cell denies it a crucial biochemical survival signal, specifically CD40L.
CD40L.
Right. Without that continuous signal, the B cell's internal clock just runs out, and it initiates programmed cell death, apoptosis.
Oh, wow. So when we take a snapshot of the germinal center and see all those weak cells, they are being permitted to live because the system is forgiving?
No. They are dead cells walking. Because the somatic hypermutation process is constantly churning out new variants, new weak cells are generated every single hour. When we look at the population, we are simply catching those weak cells in the brief terminal window between the moment they acquired a bad mutation and the moment they starve from a lack of T-cell survival signals.
It is dark. The boot camp isn't soft at all. It is absolutely ruthless. We were just completely misinterpreting the photograph.
Exactly.
So recapping this final section, 10-fold affinity means daily doubling, capping at 40 picomolar. And the diversity we see is just an illusion caused by survivorship bias and doomed cells, which makes you wonder, where else in our daily lives might we be completely fooled by survivorship bias when looking at data or studying success stories?
It's a great question. And when we pull all of these complex mechanisms together, a really cohesive picture of the immune system emerges. Germinal centers are incredibly noisy and messy environments. They generate wildly disparate evolutionary trees. They are fundamentally hobbled by the spelling biases of their own DNA, routinely missing the most effective upgrades.
Right.
And the physical space is just filled with doomed cells walking toward inevitable apoptosis. Yet, despite all this profound inefficiency, the brutal, persistent selection process reliably drives the population toward a higher affinity. It works.
It is a totally messy, chaotic system, but it keeps us alive. Which leads to a really provocative thought I want to leave you with today. We just spent this entire deep dive learning that natural immune systems leave a massive amount of binding power on the table. They physically cannot access certain incredibly potent mutations because they are locked out by the DNA lottery and the aid enzymes blind spots.
But now, with technologies like deep mutational scanning, we possess the comprehensive map. We can identify the exact molecular upgrades the B cell is forbidden from making naturally.
Right. So consider what happens when we use this map to actively engineer vaccines or therapeutics. If we know the precise molecular tweaks that nature is blind to, could we just bypass the DNA lottery entirely?
Could we synthesize monoclonal antibodies in a laboratory that are fundamentally superior to anything a natural human evolutionary process could ever generate on its own?
Exactly. We are standing at a threshold where we transition from merely observing evolution to actively outsmarting its inherent constraints.
It fundamentally changes our entire approach to disease management and therapeutic design.
It absolutely does. Well, thank you for taking this deep dive with us. Remember, the next time you recover from an illness, there wasn't a clean, predestined path to your recovery. Inside you, there is a chaotic tape of life, constantly rewinding, playing again, and finding a beautifully messy way to win.